Hello students, today we'll start with the topic biotechnology. As we have discussed earlier about the aspects of biotechnology and its application in different fields. Now, today the most important we are going to discuss is about the technology known as recombinant DNA technology. Very popular technology used in the field of biotech. Recombinant means a recombinant desired a variable parents are taken and they are crossed and the new product called recombinant is obtained and the changes occur in the DNA. That's why recombinant DNA technology, also called as gene cloning or DNA cloning. So first we will describe what is recombinant DNA technology. Recombinant DNA molecule is produced by joining together two or more DNA segments that is obtained from the different organism. That means the desired DNA molecule of our interest, we can isolate that DNA segment and from the different organism and can make a recombinant molecule. Now thing is how this process is done. So for this we use specific enzyme for cutting the DNA into suitable fragment and then joining in a suitable host through a specific vector. Now the next thing, the two things we have discussed here is the enzyme for cutting and the suitable host to transfer and the medium is vector. Now second, the DNA fragment are inserted into another replicating DNA molecule and that molecule is known as vector. And primarily we use this, for this vector we use a bacterial, very good source, a bacterial plasmid. Now the thing is, what is plasmid? If we discuss the structure of bacteria, this is the structure of bacteria. This is the main chromosome and beside this main chromosome we have a plasmid. This is the plasmid. This plasmid is a closed circular extra chromosomal DNA and this one is the main chromosome, that is bacterial chromosome. Now this, when we prepare this vector by inserting a desirable DNA, now this vector primarily used as we use a plasmid Then this vector is introduced in a particular organism called host. This is the bacterial cell, and the whole process, the transferring of vector into the host, is known as transformation. Now, our desired DNA segment will be obtained in multiple numbers, and this whole process is known as DNA cloning. For a particular DNA of interest, we get a number of copy exactly same for a gene cloning. Next, we'll discuss step by step the process of cloning. So, steps in gene cloning. Here are various steps in gene cloning. First is the production and isolation of DNA fragment to be cloned. Next is this isolated DNA fragment is then inserted into a suitable host to obtain recombinant DNA. Next, this suitable recombinant DNA is then introduction of recombinant DNA into suitable organism, that is called as host, and the process is known as transformation. Next, fourth is the selection and selection of transformed, that is recombinant host cell. That means the transfer host cell in which we get the desired DNA in the recombinant form. Host cell and identification of clone containing desired gene or DNA fragment. Next is the multiplication or expression of introduced gene into host. So this is the step-by process of gene cloning. Now next we'll discuss the graphical representation of this process. First, we have to take the foreign DNA, the desired DNA, which we have to need to express desired DNA. Next, we have to choose the vector which we use is the plasmid, which is a circular covalently closed. This is the vector DNA, that is plasmid. Now we can cut the vector and insert the desired DNA into it. For this, we need a restriction enzymes. Restriction enzyme cut both DNA segment and vector DNA at specific site. Now, the specific DNA at the cut site, the restriction enzyme cut that, and the covalently closed vector in which the DNA, desired DNA has to be ligated. This is the desired cut DNA fragments. This is our vector. Now the next enzyme used here will be the DNA ligase. Ligase is the enzyme which join the DNA. So here the DNA ligase will join desired DNA into vector. Now this is a ligated desired DNA with the vector. This molecule is known as recombinant DNA. This now transformed into host cell. And the best host we have discussed is our bacterial cell. This is the main chromosome. And nextly, this is the plasmid we are isolated for the expression of our desired DNA. And the bacteria we used here is the E. coli, that is Escherichia coli, as a host. Now, as the bacterial cell will divide, it will generate a number of bacterial cell. As the number of bacterial cell will increase, our desired DNA within the vector will also increase. This is the process how the gene get transformed and multiplied, and we get our desired DNA. So this is all about the this class. In the next class, we'll discuss the tools of gene cloning in detail, that is restriction enzyme and the DNA ligase, and further examples of restriction enzymes and new techniques like gel electrophoresis used in the biotechnology. Thank you.